Just in summary, E. coli producing the CTXM enzyme are relatively rare in coastal waters, but levels can be high enough to pose an exposure risk to certain water users, such as surfers. And we found that there was a statistically significant association between surfing and gut colonization by these CTXM-producing E. coli. So we think that studying gut colonization is a really interesting and important outcome because although these surfers and their controls are likely to be asymptomatic, harmless bacteria colonizing their gut, as we heard from Matt, people can share their gut microbiome quite easily. Gut bacteria are often a major source of infections, opportunistic infections. And like I said, if they spread to, say, vulnerable members of the population who could go on to develop drug-resistant infections, this could be a real problem. We did look at other antibiotics. We also looked at bacteria that were resistant to meropenem. And we did find it in surfers and non-surfers, but at approximately similar ratios. Recreational exposure to antibiotic-resistant bacteria in coastal waters is likely to be much greater. Cefotaxime is, what do you call it, you call it like a big boy antibiotic, or whatever you called it. Sorry. Things like ampicillin might be a much more common resistance phenotype in bacteria. So recreational exposure, we might find much more of those kind of antibiotic-resistant bacteria in coastal waters. So exposure might be much greater. But certainly coastal waters could be a more important transmission route than previously thought. The final study that I'm going to talk about involves using animals as kind of sentinels for resistance in the environment. So these small mammals, unlike humans, we can trap them at their location. Because they have small ranges of movement and migration, they're likely to reflect the environmental conditions that they're actually in. This is actually a published paper that was published earlier this year by one of our master's students, which can be found in environmental research. Essentially, we live trapped small mammals across 7 coastal sites in the southwest, and this is downstream of a wastewater treatment plant, which serves a population of about 25,000 people. Each coastal site was paired with an inland site, which was about 100 meters away, and these small mammals were trapped, and then we tested their fecal matter for E. coli that were resistant to this panel of antibiotics, affecting a few different classes of antibiotics, but we've got cefotaxime in there, ampicillin, ciprofloxacin, and trimethoprim. As this graph shows, the mean proportion of small mammals carrying E. coli resistant to these antibiotics is much greater in the coastal mammals compared to the inland mammals, and this tends to be significant. If you look at all different types of AMR in E. coli, you can see that the risk ratio is about 2.3, which reflects quite well what we saw in the surfer study, the risk ratio of 2.95. So when we kind of weigh up this evidence and consider what more do we need to kind of demonstrate a causation, I've ticked the ones where I think that we've demonstrated that we found evidence in terms of we found an association between exposure to coastal environments and gut colonization by resistant bacteria. We found it in both animal studies and human studies. If there's kind of biologically plausible ingestion, we know that ingestion of contaminated drinking water, for example, can result in illness and in gut colonization by resistant bacteria. And we've observed that in coastal waters, your bathers are much more likely than non-bathers to have infections or symptoms of infections. So I really like this quote from the Sir Austin Bradford Hill paper from 1965. I'll just just because all scientific work is incomplete, and just because we haven't ticked off all of these kind of lists, these criteria on the list, it doesn't mean that we should say that it's not causal or it is causal. We just need to, something as important as antimicrobial resistance, I think that we need to kind of take these bits of evidence quite seriously, and kind of, I think this is really important for us to take action, particularly when it comes to pollution of our environment and looking after our environment. Humans have a huge amount of interaction with their natural environment. It has many health benefits as well as potential risks. So we need to look after it, and that's it. I think looking after the environment will kind of produce a multitude of co-benefits to human health and well-being. I'd like to acknowledge all the help that I've had from my colleagues at the University of Exeter, as well as those outside the University of Exeter, Professor Peter Hawkey from Public Health England, Dr. Jonathan Porter from the Environment Agency. This work wouldn't have been possible without the participation of the Health and Environment Public Engagement Group down in Truro, who gave us feedback on our proposed methods and how acceptable it was to ask people for a rectal swab, as well as Surfers Against Sewage, who obviously helped with our recruitment and communicating with surfers, and all of their support involved in actually getting samples. I'd also like to acknowledge our funders, the European Regional Development Fund and NERC. Right, that's me.
Thank you very much, Anne. So Anne sort of very nicely sort of highlighted the importance of considering the environment when we're trying to talk, think about strategies to sort of slow down the antimicrobial resistance problem. So, without thinking about the environment, then really sort of leaving a big area and sort of unthought about. So, any questions for Anne, please? There's a mic coming, John.
How representative are the rectal swabs of colonisation? I mean, are they pretty good, or are we missing quite a lot of cases?
I did actually pose this question because obviously the gastrointestinal tract kind of is quite large, and what is at the bottom of it might not be what's going on at the top. So, if the swab indicated that there wasn't an infect, like colonisation in the lower gut, it doesn't mean there's not colonisation higher up, and vice versa. So I think it probably is a good indicator, but it's not a perfect one.
Can I ask about the positive cases you had? Can you study family members? So you've got a small cohort of positive findings. Will your study look at family members?
We finished this study at the moment, but it's something that I'm really interested in looking at, because we could be seeing that there's a transmission from the environment to human to family and family groups. So we don't know whether this colonisation stops at the surfer or whether it goes off to other people.
Do you recommend I give up the surfing?
No, I enjoy, well, I can't surf, but I try to bodyboard every now and then. I think there are lots of health benefits from going out into the sea and enjoying being in the sea. I would also say that there's no evidence yet that you yourself are going to get sick from an antibiotic-resistant infection. I mean, this is plausible. The evidence from previous research has shown that it's possible, but we just don't have a large enough study to know whether there are any long-term health effects and negative health impacts of having your gut colonized by cefotaxime-resistant E. coli.
Was there a geographical distribution about the existing organisms? Did you find that there was any correlation between those and the proximity to the sewage outflow or to runoff from agricultural fields?
So with our sample size, we got, 9 people were colonised by, 9 surfers were colonised by CTXM producing E. coli, which isn't really a large enough sample size to draw any meaningful conclusions from, but we'd love to do a bigger study, obviously. We recruited people from all over England, Wales and Northern Ireland. Most of our participants were from the southwest, and we did ask people, in part of the questionnaire, we asked them about risk factors we thought might be associated with gut colonisation as part of surfing. So how often do they go surfing? How long do they spend in the water? And things like, do you look up water quality at the beach that you're going to go to before you go? And if you do you pay any attention to it, or do you just say, well, surf looks good, I don't really, this might be a warning up, but I don't really care, I'm going to take the risk. Again, because of the small sample size, 9 people colonised, we couldn't really draw any meaningful conclusions from it, I'm afraid.
Got a question down here. Okay.
There's a question over whether farm animals actually contaminate the environment, and that might lead to persistence of resistance even though blood control took place on the farm. Questions about whether wild mammals could transmit resistance on the farm more widely, flood farms to people, etc. Have you done any work, or has anybody done any work?
Well, when it comes to water quality, there is a bit of work being done on microbial source tracking. So using markers to see where these bacteria, whether they're kind of sheep-associated, cow-associated kind of thing. We haven't done that with our bacteria. What we did do is we did the multiplex PCR to identify the phyla group that each E. coli belonged to. So E. coli is quite phylogenetically complex. And I think you showed some little, in your family diagram, showed some different coloured lines between different family members which represented different phyla groups. We found that the majority of these ones that was harbouring CTXM were a human pathogen called ST131. It causes urinary tract infections. It's quite commonly associated with CTXM carriage as well. So we weren't just finding CTXM bearing E. coli and commensal bacteria. I mean, they were present, but we also found ST131, this pathogenic E. coli, in fecal samples from surfers as well as non-surfers, which I suspect is probably human source, I would have thought. Although I don't know what work's being done on ST131 in animals, I'm afraid.
Were there any of the samples, were anybody actually infected in some way or other at the time of the survey or prior to it?
We didn't screen people for kind of infection before, and we didn't ask people about their current health, I'm afraid. But in a different survey, I have looked at self-reported symptoms of infection by surfers and swimmers. Perhaps we can talk about it at the end, afterwards.
First of all, I'd like to congratulate you. Surfers are generally very tight-fisted group. And the fact that they gave you their gut bacteria for nothing should be applauded. But secondly, regarding your mammals of origin and coastal zones work, did you notice any difference regarding the technologies that were employed at the water treatment works? For example, if they were UV treated water?
My goodness, I can't remember that. Yeah, that's a really good question. I might need to go back to the paper and reread it. It's not my paper, I'm afraid, but yeah, could be. Yeah, I'll have a look.
Any more questions? Yeah.
Hi. You mentioned briefly about the general health of the people that you took into the sample. Did you look into their diets and whether they had a significant amount of probiotic included in their daily intake, and whether that would have made a difference?
Oh, that's a really good question. We did ask about diet, but it was mostly related to consumption of different animal products, so meat, chicken, pork, beef, seafood and things like that. We didn't actually ask about probiotics, which could be really interesting because that could maybe prevent infection. But I do wonder with those whether, we're talking certainly about CTXMs, we're talking about a mobile genetic element which can be passed from an invading E. coli. It could be passed to your gut bacterium, it could stay in resident there and then pass it on to a pathogen. So I don't know how protective probiotics would be against colonisation. I'm not sure.
Okay.
Hi. Can I ask, have you considered extending your work to look at usually doctors at Cornwall Hospital? Most of them seem to spend a lot of time surfing. It's quite an interesting point from everything else because we should have a big problem with resistant E. coli in the world.
I hadn't considered that. I didn't know how willing junior doctors would be to. I mean, my experience with the surfers was quite difficult. I mean, it took several months to get 150 swabs.
I think they'd be up for it.
They'd be up for it. Yeah.
Especially if they've got senior clinicians, sort of,
Yeah, it would be interesting to know what the flow is. Is it? It'd be interesting. Yeah.
You need to prove clinical relevance of what you do is great, but you need to show further down the line it has some significance.
Yeah. So there's surfing doctors. Yeah. It would be interesting to see the direction as well, whether it's from environment to doctor to patient, or patient to doctor, or patient to C. And, yeah. Sounds messy.
Anne, you are looking for sort of another research idea, aren't you? So
Yeah.
I'd be happy to sort of explore that. Any further questions, please? Yes, Sophie. If you stick your hand up, have you seen him do that? Yeah.
When you selected your Environment Agency coastal waters, did you use just the bathing beaches, or did you use the streams? Because the prevalence of E. coli in those streams is way higher than it is in the bathing beach waters, and it's probably a much greater population and risk. Did you use the actual sea waters, or did you use the stream data?
That's a really good question because, like you say, E. coli abundances in stream water tend to be much higher. We use the stream water samples because we wanted to express it as a prevalence, and we're much more likely to detect the presence of it in slightly dirtier water samples, but still if you apply that prevalence to coastal water from the bit that people actually go bathing in, or swimming or surfing, you should see approximately the same absolute numbers of resistant E. coli. But also people do go in the streams, certainly young children and their dogs play in the streams because it's seen as a kind of a safer environment. But yeah, that's a good question, thanks.
We've got time for one last question, one burning last question. Okay, thank you very much, Anne. Thank you. Thank you, that's great. Perfect.